Biological molecule, nucleic acids. The two examples are RNA and DNA. The monomer is a nucleotide. The nucleotide consists of a nitrogen base attached to a sugar, which is then linked to the phosphate. Note the phosphate is linked on carbon number 5 and the nitrogen base is linked at carbon number 1. When we look at nucleotides, we'll start off with a sugar. If you are RNA, the sugar is a ribose sugar, and attached at carbon number 2 is this hydroxyl group. If you are a DNA, then the sugar is known as a deoxyribose, and instead of a hydroxyl group at carbon number 2, you have just a hydrogen. This is an important difference between RNA and DNA. When we start building our nucleotide, we first start by adding a base to the sugar. The base is added at carbon number 1, so it's a glycosidic bond between the sugar and the base. There are 4 different types of bases in DNA and 4 in RNA. These bases are divided into purines and pyrimidines. Notice that in RNA, the pyrimidine is uracil, versus in DNA, it is thymine, and the difference is the methyl group that you see attached on the thymine. Otherwise, they are identical molecules. The other thing to notice is your purines have 2 nitrogen rings, versus only 1 nitrogen ring in your pyrimidines. Guanine and adenine are purines. Cytosine, uracil, and thymine are your pyrimidines. The third part of a nucleotide is the phosphate. The phosphate is attached on carbon number 5 by a phosphodiester bond. And you can see that here are your 3 phosphates. 1 phosphate is called a mono. If you have 2 phosphates, it would be a diphosphate, and 3 phosphates would be a triphosphate. So when we're building our DNA or RNA molecule, these phosphates are actually what help give energy to form the bond. And you can always only make DNA by going in the 5 to 3' direction. So that new phosphate would actually come and be attached on the 3' in so that you would continue in the 5 to 3' direction. So when you finish forming your DNA molecule, you can see this phosphate attached to the sugar, attached to the phosphate, attached to the sugar. On the opposite strand, here you can see 5' phosphate, 3' attaching to the next phosphate. If you notice the position of the oxygens, they are opposite on the 2 molecules. And then you see how the bonding forms between the bases. The bonding is hydrogen bonding. Thymine always pairs with adenine, single ring to double ring. Guanine always pairs with cytosine, double ring to single ring. Notice there are 3 hydrogen bonds between guanine and cytosine, and there are only 2 hydrogen bonds between thymine and adenine. Looking at this molecule, what could you guess is the overall charge on the DNA and why? And the hint would be to actually look on the outside on the phosphate. When you start seeing the actual structure of DNA, it's actually a double helix, and it's actually twisted around in a helical shape. It's called antiparallel because you can see that the strand on one side is 5' to 3', and then the opposite side is going 3' to 5'. Again, showing the base pairing. Finally, the difference between RNA and DNA is clearly summarized up in this slide. You can see RNA is single-stranded versus DNA is double-stranded. RNA has the sugar ribose. And finally, RNA has the base uracil instead of thymine. And you can clearly see the double helix of DNA versus the single strand of RNA. So this is a quick discussion of the biological molecule, the nucleic acids.